We're now making a transition in the course from the material of the first half, which is quantitative genetics within populations, to the material in the second half, which is quantitative genetics across multiple species in phylogenies. And at this point in the course, you have to be cautious, because what we promised you was a course about the connection between quantitative genetics and phylogenies, matters on small and large scales. But when you go from the one to the other, you go through a time scale. You go through time scales which jump suddenly from generations to millions of years. And it's always a bit worrisome as to whether the phenomena causing multiple species differentiation really are exactly the same as the phenomena causing generation to generation change that you can see in studies of individual individual populations over modest periods of time. So I guess the first thing I'm going to say to you all is be skeptical. Be skeptical of what you're saying, what we're saying, because we're alleging these quantitative genetic phenomena are the right thing to study, but perhaps we're wrong about that. And I think you'll find some difference of opinion among the different lecturers in the course as to how tight the connection is between the two levels. I think if you ask, some of them will say, well, we're going to use things like Brownian motion, which I'll talk about in a moment, as a approximation to genetic drift and varying natural selection. And others of the very instructors in this course are going to say, well, it's an okay model, but its phenomenology, it's a phenomenological model. We don't know if it's genetic drift. We don't know if it's variable selection. It's just something that roughly behaves like Brownian motion. That makes the connection much less tight. So I guess unfortunately, we have to issue this government warning, put a label on the course saying we're not really sure we've connected these two levels. Nevertheless, I'm going to go straight ahead and act as if they can be connected. Now, what we have to start out with is a model that we can use, a statistical, probabilistic model that we can use to model changes of quantitative characters, of multiple quantitative characters in populations. And that means we're going to have to incorporate phenomena like genetic drift of alleles, also mutation to new alleles, bringing new alleles into the population, varying natural selection, which will cause the different populations to move in different directions and individual populations to wander back and forth. And the selection could either be at the level of selection on individual loci that averages out to produce an effect on the quantitative character, or selection on the phenotype of that very character. It isn't necessarily true that the character that you study is the one that natural selection sees. That's an issue that I don't think has been raised very much yet in the quantitative genetics literature. We've tended to act as if the characters that we see are the ones under natural selection. Let me, let's see, I'm trying to, when we start out trying to use Brownian motion as a model, the people who first did that were Luca Cavalli-Sforza and Anthony Edwards in the 1960s. And they were doing work which we now see is pioneering work in development of methods for inferring phylogenies. They were actually not working on different species. Luca Cavalli-Sforza had a set of data that were gene frequencies of blood group polymorphisms in individual villages in northern Italy, in the Parma Valley. And Anthony Edwards, who was doing a sort of postdoc with him at the University of Pavia, was looking for ways to approximate the changes of gene frequency by Brownian motion, by genetic drift. And he came up with the idea of doing it by Brownian motion. And this isn't necessarily an uncontroversial choice. What you have when you have genetic drift is that the average change of gene frequency under pure genetic drift is 0. That is, the gene frequency is equally likely to go up as down on your scale of gene frequency. But the variance with which it moves changes in different parts of the gene frequency space. It dies away to 0 as you get to loss of the allele, and at the other end, it dies away to 0 as you get to fixation of the allele. So you have a non-constant variance, which the Brownian motion process that you're using to approximate gene frequency change doesn't have this non-constant variance. It has constant variance, and that's part of the reason that it turns out to be tractable. Here's an idea. Here is a calculation of the distributions of gene frequency under genetic drift, and this is in a population with a population of size 50, so there are 100 copies of the allele in this population. If you start out with 0.5 as the gene frequency, right in the middle of the scale, and you let genetic drift go on in an idealized population for 10 generations, the gene frequencies in different replicates will spread out up and down. By the way, can I ask you, you can see the cursor when I move it, is that correct? Yeah. Say again.
But maybe it would be better if you did the full screen projection instead of having the slides ahead on the left side.
Okay, well, they were partly to help me. Is that better?
That's better for us.
Okay, I can try to...
Don't overlap the screen.
Yeah. Okay, I'll see what I can do here, and I'll try to make it a little bigger, but I'm not sure it gets bigger on your screen. Is that okay? So this is an exact calculation of all of the probabilities using what's called the Wright-Fisher model, the basic model of genetic drift in natural populations, and what you see is that the gene frequency spread out in what looks like a normal distribution, although it's bounded at 0 and 1. And if you look closely, you'll see there's a tiny little bit of piling up at 0 and 1 under this genetic drift. But if you approximate it by a normal distribution, which would be the normal distribution would be the black histogram. The purple circles are the exact calculation for genetic drift. You see that it's doing a pretty good job, and you might say, okay, Brownian motion will work as a model of this. However, if we go to, and if you keep going, the gene frequencies start to pile up at 0 and 1. And although you can approximate the stuff through the center by a Brownian motion, by a normal distribution, which is what a Brownian motion would give you, it then gets chopped off at the tails. If you take a population and instead of starting at 0.5, you start at 0.1, then the approximation is not so good because you start piling up at the left side very quickly. Some of the genetic drift gets to loss of the allele, and the distribution becomes skewed, and the approximation is not as good. And if you keep going, it gets even worse because the true process piles up at 0, and the normal distribution approximation officially would want there to be mass out below 0, which of course it's impossible. So Brownian motion is a somewhat controversial approximation, but let's go with it. Let's assume it's okay. And you may say, well, why not use the exact equations of population genetics instead of Brownian motion? And the answer really is because using those to compute transition probabilities up the branches of a tree doesn't work. It's not really tractable. So I won't go over all the details of that. So we're sort of stuck with Brownian motion. Now, then you might ask, okay, you're talking about a single locus. What about a quantitative character? Well, in our simplest models, a quantitative character is a sum of contributions from a number of loci. And you could say the individual loci are approximately changing independently, roughly at linkage equilibrium at different loci, and they're changing, each one is changing by this approximate Brownian motion. And in that case, if the character is a sum of contributions from different loci, this will be a linear combination of Brownian motions, and it is, again, Brownian motion. And you can make this more or less work for multiple alleles, too. You start putting in things like interactions among alleles, actually even dominance, but interactions among different loci. And then you have to start waving your hands, which you do like this. So epistasis and dominance will cause trouble. I think I'm the first person to have taken the Cavalli-Sforza-Edwards Brownian motion approximation to gene frequencies and extended it to quantitative characters. And then if you throw mutation in, again, you can wave your hands even more vigorously. I think what we never actually had but should have is some computer simulations of all this to make sure it's working. But if it doesn't work, we actually are stuck and we have no alternative at this point. We are going to be hearing about, in addition to Brownian motion, the Ornstein-Uhlenbeck process. I think Steve may have mentioned that as an approximation to natural selection on an optimum. And that's a cousin of Brownian motion and is also tractable. And we can make that as an approximation. It has the same problems as Brownian motion. So Brownian motion is mathematically tractable. You can compute transition probabilities. There's all kinds of mathematical exotica you can talk about short-term changes in Brownian motion. But actually what we want is to compute the probability that you go from the value now to the value at the end of a single branch on a tree, on a phylogeny. And that's easy with Brownian motion. It's a sum of little wiggles, each with equal variance and each independent of the other. The changes in successive time periods are independent. And the result is that the net probability of change on a branch is drawn from a normal distribution with a variance proportional to the length of the branch of the tree. So everything works out nicely, and you get normal distributions for the change on individual branches of the tree, and then you can put all of that together. A very important point that we're going to see you make a lot of use of is that if I have two lineages and they start out at a certain value, 6.2, and then there's a period of common ancestry in which the common ancestor lineage changes, it wiggles around and ends up at 8.6. And then after that, the two species go their separate ways. There's a speciation event during which nothing is assumed to happen. And the individual resulting lineages then change independently. You end up with tip values in the two descendants, and the tip values are correlated with each other. They're each normally distributed around their original starting point, but they shared the common ancestor period. And the result is the covariance between the two tips in this single character is the variance that is expected to accumulate during the period of shared common ancestry. So the more common ancestry, the more they covary, and that gives you a pattern of covariances that matches the phylogeny. So here is a tree, and it has 7 species. And we can imagine a character value starting out at x0, and then it's going to change on each of these lineages. And the result is going to be 7 different species, each of which has a different value for the character. but those values are covarying. The expected change up any one lineage, if you go up the leftmost lineage, for example, you expect a change of 0 during the branch because the individual generations have an expected change of 0. But the actual change varies around that with a variance that's proportional to the length of the branch. So we've indicated this here by having the branch length be proportional to the expected amount of variance that you'd get on that branch. When you get to X9, you have different value from X0, and then there's a speciation, and in each of these two lineages, the change starts from X9, so they share a common starting point, and then it's independent in each of those lineages. So we're assuming the change in simultaneously existing lineages in different species is independent. We're not having any process of interaction among their phenotypes. Things like competitive character displacement would make them non-independent, but we're just assuming they each simply undergo Brownian motion or some form of variable selection. And also we're seeing that changes in successive periods up a single lineage are independent of each other, given that the second one starts from the phenotype resulting from the first one. So you put all that together, what you get is that the net change from X0 to X1 is from a normal distribution. The net change to X2 is from a normal distribution with a slightly different variance, a different sum of variances, and it's correlated with the value of X1. So X1 and X2 show a covariance, which is in fact the variance that you expect to accumulate going from X0 to X8. And that holds throughout the tree. All of these individual branches are independent, but the pattern on the tree tells you which ones start from the result of which other ones, and everything works out. And the tips are then multivariate normally distributed with covariation. So let's see, here's a covariance matrix for the tree I just showed you. And you have to go back to that tree to see that the variances that accumulate on the diagonal here are in fact the proper sums of the branch lengths on the tree. And the covariances off the diagonal are the variances that are expected to accumulate in the shared period of common ancestry. So once you can model the whole process by Brownian motion, you get a nice multivariate normal distribution whose pattern of covariances you can read right off the tree. And that's enabled all of the subsequent work that's been done using Brownian motion approximations. I should say with Ornstein-Uhlenbeck processes, it gets a little more complicated, and I'm not gonna go into that. You'll be hearing much more about that from Marguerite, for example, Marguerite Butler later in the course. Okay, I want to show you a simulation. This is not real time, but we're going to start from, this will be a 5-species tree. We're starting from the point at the bottom here. We have a speciation, and then each of the two lineages undergoes a simulated Brownian motion. I simply drew a lot of little normal steps and added them up, but you see the individual steps here. And now you get to 2 species, and we're going to have speciations occur there. So we've had them on both the left side and the right side. Notice they're at different heights because different amounts of variance have accumulated up these lineages. The vertical scale is not necessarily time, it's accumulated variance. And that might be different in different populations. So again, you get two lineages on the left, 2 lineages on the right that are very tangled. This one here is going to again be the common ancestor for another speciation. And now we have 5 tips. It's hard to see, but 1, 2, 3. Let's see. Let me go back because I want to see which is which. Yes, okay. It's 1, 2, 3, 4, 5. Now you may look at this and say, well, that's very noisy. And it is. They're really, it's not easy to just stare at the numerical values and figure out what the tree is. You need, if you wanted to infer the tree, you would need a much larger number of characters to work with, and they'd have to be relatively independent. But we're not actually going to be inferring the tree here. We're going to be using a tree and then interpreting quantitative characters on that tree. Let me see where we're going.
Joe.
Yeah, this is a very good moment for a question. Let me see if I can get the, hold on, let me see if I get the room video. Okay, here we go. Yes, please.
Yeah, I have a question about the tips. Do they need to finish at the same time that the y-axis is in time? So the last point should be alienated at the end of the screen?
I'm not entirely sure I understand the question. The vertical scale here,
I think mine is related.
Is not time. And we're accumulating. Remember that on the original tree, I'm going to go back here. We had a variance that is expected to accumulate in each branch, and those don't have to result in a clock-like tree. So the height above the original fork here, the ancestor, the common ancestor, up this lineage is V9 plus V3. Up that lineage, it's V9 plus V8 plus V1. And those don't have, V3 doesn't have to equal V8 plus V1. We're assuming that you somehow know branch lengths for these branches, and we're going to be assuming that the change of the quantitative character is proportional. The accumulation of variance is proportional to those branch lengths that you've estimated from, for example, molecular data. Does that have anything to do with your question?
Yeah, thank you.
Okay, good. I'm glad that I somehow, in all that, managed to answer the question. But I'm not sure how I did it. Okay.
I have a related question. So
yeah.
If the y-axis is variance, what's the x-axis?
Oh, the x-axis is the character value. So we're measuring a single character here, and that's the x-axis. Now, and it's true that when I draw the tree here, now at this point I'm just using an x-axis that keeps the tips separate. It's not really the character value, because at this point we don't know what the changes are going to work out to be. But here we're assuming horizontal value is the character. Vertical value is a sum of branch lengths, but not necessarily time. Okay, so let me just say before we wander on that the mode that we'll be working in here is not to take these characters and infer the phylogeny. That, of course, has been what a lot of people working with phylogenies want to do. And there used to be debates between people over whether quantitative characters, say morphological characters, particularly ones measured quantitatively, how good they were compared to molecular characters for inferring phylogenies. There are whole symposium volumes that are doing that. However, what's happened since then, since the 1980s, when those symposium volumes were published, multi-author volumes, we now have such a volume of molecular data that really there is no argument. The amount of information you can bring to bear from morphological characters is much, much smaller than what you can get from molecular data. And it's only in species where molecular data is not available, such as fossils, that the issue of inferring phylogenies from measurable characters is still very active. So most of the time in anything we'll be doing in this whole period course, somebody will have done a molecular study and will be providing you with a single point estimate of the phylogeny, a single phylogeny, or they've done a Bayesian or bootstrap study, and they provided you with a cloud of estimates. So you might have 500 phylogenies randomly sampled from the posterior distribution of phylogenies. And any analysis you're doing will be using the single point estimate of the phylogeny, or will be using the individual estimates in the cloud. And in order to take the uncertainty about the phylogeny into account. But the one thing we won't be doing is using these quantitative characters to infer the phylogeny itself. That's a fascinating topic, but it's really, it makes life a lot easier if we can work in this mode. So let us see where we are. Okay. I want to point out an algebraic convenience that comes up when you use Brownian motion. If I take a phylogeny and I take two adjacent tips, two neighboring tips, for example, tip 1 and tip 2 in the phylogeny that I just showed you earlier, and if I were to take the difference in the quantitative character between them and a weighted average, weighted with some particular weight, which I'll just call A here, if I choose the weighted average properly, if I know the branch lengths, and we'll call those V1 and V2, that they're the proportional to the accumulated variances that we expect. If I choose the weights inversely proportional to the branch lengths, so the weights have to add up to 1, so we take the inverses of the variances and we divide by the sum of those inverses. We get weights, and it has a very interesting property. It has the property that the weighted average is independent of the difference. And also, the difference between x1 and x2 is independent of everything else on the tree. This is a fact that I discovered when I was doing my PhD thesis in 1967 and published for the first, it's in my thesis, but the first publication is in 1973. And the weighted average that you compute then behaves like a tip. It has covariances with the other species on the tree. And it acts just as if you've taken those two tips and pruned them off the tree, and leaving behind a weighted average whose variance you can compute. And let me show you perhaps a little diagram of this. Here's a 4-species tree. If we take x1 and x2, we can compute the difference, x1 minus x2, and a weighted average, the weighted average being computed with these variances, v1 and v2. What we then in effect get is a little 2-species tree and a 3-species tree left behind, where this pair of values has now been replaced by an average, and that number behaves as if it's on a 3-species tree, where everything is the same except in this branch a little bit of extra branch length has been added. And here are the formulas for the weighted average, that's this one, and the little bit of extra branch lengths. And the interesting thing, property that has is that you can take a tree and you can pull a couple of species off, leaving an average of them behind. And now you have a 2-species tree and a completely independent 3-species tree. Now you can do that again. You can compute the difference between x1, 2, and x3, and a weighted average of those based on these branch lengths here. And then you go to another 2-species tree, and what's left behind is a 2-species tree. So now what you've done is factorize the tree, and it's equivalent in the sense that the likelihoods are the same. The likelihood for the original tree is now a product of likelihoods for these little 2-species tree. Well, this is just mathematical niceties. What good is it to us? What it does is it lets you come up with quantities that respect the structure of the tree. They're computed using the structure of the tree, but are now independent of each other. And we'll see when we do comparative methods in a moment that becomes important to one particular approach, which is called the contrast method. So let me now talk for a bit about where natural selection comes into all of this. And we start out with Russ Landy, as happens with so much in this course. And we also start out with the Breeder's equation, which I think we saw earlier. I mentioned it in my quantitative genetics lecture, which you'll recall is the classical result that the change of a character is equal to the product of the heritability times the selection differential. And as you've been told by Steve, Russ Landy managed to take the selection differential, which is the difference between the selected individuals and the original population mean, the mean of the original population from which they were drawn. And Russ pointed out that when you have a fitness surface, a mean surface of mean fitnesses, so it would be the adaptive topography that Steve talked about, that the selection differential is equal, this is just a version of what he wrote, to the phenotypic variance times the derivative of the log of the mean fitness with respect to your character. So when you have an adaptive surface, we can use a version, a sort of Landyized, a version in Landyland of the original Breeder's equation, and it comes out, of course, as the G matrix, which here is called V sub A, times the derivative of the log of mean fitness, the slope of the log on the surface of mean fitness. So, okay, that's the Russ Landy review. And the result of that is that when you have selection towards an optimum, here the optimum is Gaussian because that makes life easier. It's not because biology demands that it be Gaussian, but because the mathematics demands. Here is the expression for the fitness curve. And again, Steve was using, this was, I think, omega or something in his notation. And the result, of course, is that you can show that the change of mean fitness is equal to this thing, which was G over P plus omega. Steve, am I right about that? Was that your notation? Yeah, times the difference between the current mean value and the optimum. So here it's the optimum minus the current mean value. And all that results in is that you move a constant fraction that as long as these additive variances and phenotypic variances and the shape of the fitness surface remain the same, each generation you move a constant fraction of the way towards the optimum, and what that fraction is depends upon this G over P plus omega. Okay, so we remember that. Now, if that's the form, that is a typical form is believed to be a typical form of natural selection. Let's then talk a little bit about how that would work out on a tree. And how it will work out if you have multiple characters, you have a multiple character fitness surface, and you have the population trying to climb the fitness surface, but you also may have, as Steve noted, that the fitness surface itself can move. So the population is always trying to climb the surface. The surface is perhaps wandering around by some process, and sometimes we wave our hands and call that Brownian motion also. And the result will be that you have, and you also have genetic drift during the whole process. So you have wandering due to genetic drift. The process of genetic drift is affected by the genetic covariances. If the traits covary, then genetic drift will reflect that genetic covariation. Also, the response to selection is affected by the genetic covariances, the G matrix. So many people look at that and they'll say, oh, okay, the process of natural selection depends on the G matrix, and so does genetic drift. So if we see covariation of characters across the phylogeny, looking at the tips, it must be the reflecting genetic covariance. And what I really want to do is to tell you in this part of the projection is that there's another source of the covariation, and that is called selective covariation. And it's very little known. It's very little discussed in the literature, which is, I think, is a great problem. It was first introduced by I think he was Swedish, well, he's Swedish or Danish, I can never remember which, Olaf Tedin in 1926. And G. Ledyard Stebbins, in his great book, Variation and Evolution in Higher Plants, noted this and helped publicize the concept. But let me explain it to you for a moment. And basically what it is it's covariation of selection pressures. So if we're looking at a lineage and we're following the change of multiple characters, and characters are covariing, they're covariing for both of these reasons. It's the change of phenotypic means as a result of genetic covariances and these selective covariances. And let me give you a simple, somewhat silly example of selective covariance. Let's imagine a phylogeny in which the lineages end up in different environments. So this lineage is in the temperate region, but this lineage is in the Arctic, and this lineage also entered the Arctic. The result might be that as a result of, if these are mammals, there are things called Bergmann's rule, Allen's rule, and Glogler's rule, which says that if you're in a cold temperature environment, you expect natural selection to favor being large, being dark in coloration, so your fur can pick up solar radiation, and to have shorter limbs relative to your size so that you will lose less heat. So let's imagine that those rules are working, and so in these lineages that went into the Arctic, these individuals got bigger, they had relatively shorter limbs, and they got darker in fur color. Now, if you look across the species, you might say, aha, those characters are covarying with each other. They must have genetic covariance. And the point I want to make here is that that's not true. This is a result of selective covariance. There's no implication here that size and limb length are genetically correlated, though they might be, or size and color, or limb length and color. Those are simply cases where there have been selection pressures that are correlated with each other. Their occurrence is correlated as a result of the interaction between the phenotype and the environment. So I just want people to keep in mind selective covariance, excuse me, selective covariance as well as genetic covariance as a source of the covariation that we see among traits. Okay, we'll speed up a little.
Question
no, you can't. You can make a
because
comment.
we've got plenty of time. So I think you're right that this covariance in selection, I think, is not discussed in the empirical literature much, but I think it resides in the characterization of selection we were using up until today, and that is, if you think of that omega matrix, then I think what you're talking about, that kind of selective covariance, is in the off-diagonal terms of the omega matrix. It's talking about, if you think of the selection surface as being circular, that's an omega 1,2 that's 0. But if omega 1,2 is positive, then the whole surface is tilted in an elliptical way, or tilted down if omega 1,2 is negative. And I think that's the kind of thing you're talking about. What do you think?
Yes and no. If we have an adaptive, selective peak, what were you, remind me of your terminology. You had the individual selection surface and the other one you called the adaptive topography. What did you call it?
So if they're both Gaussian, this is Landy-Land result. If they're both Gaussian, then the individual selection surface is described by a matrix in which the variance like terms are omega.
Right.
If the corresponding adaptive topography, the surface is described by a matrix in which the elements are omega plus p.
Right.
Yeah.
Now, that's for an adaptive topography that sits in one place and doesn't move.
Yeah.
And we're asking about the movements of the characters back and forth on that peak. But when I talk about selective covariance, I also mean covariation that might result from the peak itself wandering.
Right.
So we're not just talking about the shape of the peak. We also have to be sensitive to the fact that the peak can wander, and if you have multiple species, you may have multiple peaks. Here, I'll try to, here are multiple peaks, and they can wander separately. So it's a little bit more complex than just the issue of the shape of a single peak.
Right. So I think we now have to make a choice for what I think of as the peak controller process. So in other words, the peak is moving, but what is the process that's describing that motion? And
yes.
if we're Hansen and et al. In 2008, then we choose Brownian motion for that. And there's a
natural choice because it's tractable.
Yeah, I think I can't remember. I have to go back and look. My memory fails me here. I think we made a choice of. I guess my choice would be to make that matrix that's describing Brownian motion of the peak. I guess my choice would make that matrix proportional to the omega plus p matrix.
Well,
I can't remember exactly what
I don't think there's any rule that says that.
There's not a rule, but if you forced me to make a personal choice, I would make it proportional.
That's because you're self-serving.
It is self-serving. I've been accused of this is Arnold's special place in which everything is.
Yeah, and
proportional to everything.
I have to say, as a biologist who's worked with theory for all my career, there is a general principle that says that the real world out there corresponds to our models, and if it doesn't, so much the worse for the real world.
I embrace that too.
Yeah, but I would think that when you're thinking about individual species following individual wandering peaks is maybe not a bad model. Then you start getting the peaks wandering, and you have to worry a little bit. You have to say, well, could they be on a common peak? Well, sort of, maybe, but most likely they're on somewhat individual peaks. What happens at the moment of speciation? Do you go from having one peak instantly to having two peaks? Do they wander in a correlated fashion for a while? And when you start thinking about that, that way lies madness or ecology. That basically it's all very well to think about quantitative genetics, but if you want to start thinking about peak movements, if you really wanted to have a model of them, you'd have to really understand how these characters fit in to the ecology and how that generates the fitnesses. And I think for everything we do in this course, we're not going to be able to do that. Maybe somebody will have an example later on. If so, they should tell us. But I think the kinds of studies that we're doing now, that any of the lecturers here are doing, have not reached that level of understanding of how the phenotypes fit into the ecology. They call it the abiotic part of the ecology and the biotic interactions as well. And so we're just waving our hands and saying maybe it does Brownian motion, or maybe it does Ornstein-Uhlenbeck process. And at the moment, that's pretty arbitrary. I wanted to
Joe, could I,
show.
sorry.
Is there
Before
another
you move
question?
on- Yeah, it's Marguerite. Hi. How is it different, what you're talking about, from convergence? Is that what you're talking about?
Convergence.
Yeah, convergent evolution.
You could have convergent evolution in a situation like this by having the peaks pulled together. So you could have a phylogeny, and there are let's say organisms are they're wandering on peaks, but they're separate peaks. If for some reason of encountering similar environments, for example, the peaks for two species that are perhaps at different places on the tree are attracted to each other, that could result in convergence. It's very important to note, by the way, that convergence is never in all characters of the species. Individual characters or sets of characters can converge. A dolphin and a tuna fish can come to have roughly similar shapes, but if you start looking at their innards, you find that they're very different organisms. The whole organisms don't converge, and there's a lot of misunderstanding of that in the public. I debate with creationists online, and creationists are always sure that entire organisms are converging on each other, and they use that to refute inferences of evolutionary trees. Anyway, it's a separate question. You can have convergence, but for the models that we'll be making here, we don't really have it in the model. I think we need to know more about the biology to know which peaks are likely to converge, have their optimum values converge on each other. Is that addressing your question?
I think I'm learning that we are looking at the same thing and seeing very different things. So that's very interesting. So your view is that each species has its own peak, basically, and not that the peaks exist in common for all the species in some way.
Oh, so when you're thinking of convergence, one of the situations the one that perhaps you're thinking of is that there is one overall peak, and all these species, although they're speciating from each other, are wandering around on the peak, the same big peak, which results in them being pulled towards the same optimum. And is that perhaps the convergence you're thinking of?
Yes.
Okay, that's a different case. These are different cases. You might have a case where there's one overall peak. You might have a case where individual species, individual lineages, because of their different ecology, have their own peaks. And I think it's early days yet for using those models. And in order to make those models real, we're going to have to model the ecology, really, the functional ecology of the characters. And most of us haven't done that. Okay, other questions here? Let me go back to this case here, which is a simulation I did just to give you a feel for when genetic covariance is important and when selective covariance is important. And this somewhat duplicates things that Steve talked about earlier. Let's imagine what I've got here is a single lineage evolving through time, and it's got genetic covariance between two characters. It's going to be negative covariance in this particular case. And what I'm showing you is a population undergoing, let's say it's genetic drift, and it's on an adaptive peak. And what we would see, because of the genetic covariances, which then affect the covariance covariances of movement on the peak. If I have all these circles here be the successive generations, we can't see which is which, but so the population is wandering on this peak, but it's tending to wander in a negatively correlated way. And you can see that in the distribution of the first 100 generations. But now let me also assume that during that process, the peak is starting to move. The peak is moving. So the peak has actually been moving. And I'm going to assume that the covariation of the means of the optima is positive, not negative. So the peak is going to be wandering up and down on this sort of mostly near the principal diagonal, the 45-degree line. So if we go forwards, if we go now, not the first 100 generations, but the first 1,000 generations. So now we plot, I showed you every generation for the first 100. Now I'm plotting every 10th generation. So there's still 100 points, but they're now spread over a bigger time scale. They're going from, generation 0, 10, 20, 30, 40. And here they are. And now what you see is that the negative correlation of positions is a bit spread out, and it's kind of the correlation is somewhat lost. If I go longer in time, if I go 10,000 generations and show you every 100th generation, you can see that in fact what's happening is that the means are more or less following the peak. So the peaks are wandering this way, and they're dragging the population along with them. Within short terms, the covariation of movement is negative. So if we look at just successive generations, they'll tend to vary on this counter diagonal, but overall, for the long period of time, the variation is positive. So this is just a trivial example of how the covariation of the optima in the movement of the peak has a very strong long-term effect. And in the end, the population is just being towed around by the peak. And the genetic variation creates short-term correlation. And I should point out also that in addition to the covariation of movement of the optima and the genetic covariation, there is the question of, well, what's the covariation induced by the shape of the peak? And that's what Steve was talking about earlier. So all of these come together, but in the very long run, it's the covariation of the movement of the peak, of the optima, that has the long-term effect. And if we're following a process of Brownian motion through time, in the end, we would mostly see movement that reflects that. So this is just a cautionary numerical example. Okay, where are we? Okay. Yeah, Steve has already talked about this a bit. I just wanted to bring up that. Oh, okay. No, sorry. This is a case that's different. I wanted to bring up a case. I mentioned that we've been assuming in this course that the characters that you're measuring are the characters that natural selection sees. And I think it's worth thinking about cases where we look at one character and we say, oh, the size of the organism. But it turns out that natural selection is assessing not that size, but some other measure of size. So you have two characters, one of which you're measuring and one of which you're not, and they're genetically correlated with each other. One of them, maybe the one you're not measuring, Y, is under optimum selection, but you're observing X. And if you start thinking about what that would result in effect, what you see will be the process of, if there's a single peak and it's not moving, you would get an Ornstein-Uhlenbeck process for Y, a Brownian motion that's constantly towed back to the optima, plus the to the extent that X differs from Y, the parts of X that differ from Y, the contributions that differ from Y, they may be undergoing Brownian motion from genetic drift. So you'll have a weighted average of Brownian motion and Ornstein-Uhlenbeck processes. I think in most of the studies that I've seen, people have not considered this, and I can sympathize because it makes life hard, and it involves characters that you're not actually measuring. But I think we have to always keep things like this in mind when we're trying to model the change of multiple characters on short or long time scales. So that process will look a bit like an Ornstein-Uhlenbeck process with a wandering optimum, but it's not exactly the same. So, okay, now if we can think about multiple characters on a tree, yeah, yes, please.
Hi. This what you just said. I'm always thinking about, there's a lot of studies that measure just body size. So you're interested in the evolution of body size.
Yeah.
And you might get some measures, some species, you have the means. And you have like some climatic variables also, means for the species. And you can do like an evolutionary regression in which you assume an Ornstein-Uhlenbeck process. And you can find a significant relation between climatic and body size, right? But at the same time, if you're used to studying with multivariate traits, you can see that size is normally a genetic constraint. Like there's a lot of variance on the size direction. So if selection is actually acting on another thing, such as shape, I don't know, most of the responses will be on size. So if you're using a univariate trait that actually normally is a constraint in the multivariate case, because you can see that PC1 is equivalent to size, right?
I missed the last phrase. You can see that what?
You can interpret PC1 as size, right? We normally do that. PC1, yeah, the first principal component, right?
Yeah.
But it is Gmax or Pmax. So it's driving the divergence to its direction. You understand what I'm saying? I'm always asking myself, how does comparative methods deal with constraints?
Yeah. Well, we haven't got to comparative methods yet. And also, I want to put a footnote on your statement about regressing characters on environmental variables, because there's a statistical problem there, which I think people aren't taking enough into account. But we will be talking about multiple characters, some of which may be size, and how they're changing on a phylogeny and trying to infer the covariation. That's really where we're going from here. And it's a good moment to raise this because I was just about to invoke some of these things. It's true that if size has a lot of genetic variation more than shape, then selection on shape can end up with a response, a large response in size. That certainly is true. I think it connects to exactly what I was about to talk about, that we would like to know the covariation of selection pressures, which might in that example be on shape, and the kinds of responses that would result, which result from the selection on maybe shape and the genetic covariances, which then translate that into changes in both shape and size. Let me just for a moment go back to the thing I was talking about on this slide, because I think it's going to relate. if you wanted to understand the whole situation, what you would do, you'd want to do in the ideal case, is to do quantitative genetics on your beasts. Now, I realize a lot of people work on things that you can't do quantitative genetics on. I don't think anyone's ever done quantitative genetics on whales, for example. But if you had an organism that you could breed in the lab, you could imagine making estimates of the genetic covariation by standard breeding experiments. And that sort of thing Pat Carter and others have been talking about here. You could also imagine somehow inferring the resulting covariances of changes on the phylogeny. So it's the covariances of the responses. And I'll be getting to the comparative method stuff in a moment, where we'll talk about that. And then, of course, Margarita is going to be making that much more real in the next lecture. Well, if you know the G matrix, and you know the covariation of the selection response, you could think about back calculating and inferring the selective covariance indirectly from the observed differences of species and from your G matrix. So it would be those selective covariances that you would have to have in order to get the results you got given the quantitative genetics. Now, this is an idealized it's an idealized program of research, and I think most people can't do it. Ideally, you'd like to have quantitative genetic experiments and get G matrices on all of your species. Nobody ever has been able to do that. But I think we're probably moving gradually towards that in some of the most favorable cases. So the points I'm making are here. You're back calculating selective covariation. And I think that in a sense, that's something that people would really like to know. It's nice to know the G matrices, but what you'd really like to know is, what is natural selection doing in nature? And how does that relate to the ecology? So, and I should also add, there are hopes that by looking at short-term changes, you might get information on G matrices. Well, if you have a phylogeny, short-term changes are what you see if you have, I can't do it with my fingers, if you have two tips that are very close to each other on the tree. So I think there is some hope. I'm just sort of waving my hands. I think there is some hope of getting information off the tree. And if you can't do a breeding experiment using very, very closely related neighbors to get one kind of information that is mostly genetic covariance, and larger differences between more widely separated tips on the tree, giving you the other kind of information, which is the net covariance of the selective, of the genetic and changes that occur on the tree. So the other possible way of getting information on selective covariances or selective correlations would be if you somehow really understood how this character's functional ecology, so that you could predict what character values would result in what fitnesses. That's, again, an idealized situation, and I don't think you'll find very many examples where people can do that. But you can hold it out for yourself as a sort of research program of the idealized future. And I think it's worth talking about these things, because in the long run, they give us some idea where the field might be going. I know this sounds very hazy, but rather than just saying, well, we infer X, Y, Z, we infer G matrices and do this and do that, maybe we want to step back and think about how all of this fits together and what insights it gives us about natural selection. Okay, so that brings us to comparative methods. in the literature in fields like zoology and botany, you used to find, before the 1970s and 80s, the mention of something called the comparative method. And senior biologists were always saying, we must use the comparative method. And the only problem that you would have is if you pinned them down and said, excuse me, what is the comparative method? What they would say would start to not make sense. But what it always involved was looking at multiple species. But then when you ask them, well, what's the method? They really wouldn't have a very clear prescription. But in the late 1970s, among biologists, mostly in Britain, people like Paul Harvey and Tim Cluttenbrock and others, there started to be a discussion of how do you use multiple species data to infer things like the evolutionary correlation between different characters. For example, the relationship between brain weight and body weight observed over multiple species. And they started to realize that there is some sort of problem here because species in a genus were correlated with each other. And so it wasn't completely clear that counting individual species as data points was the right way to go. People started discussing the classification system and saying, maybe we should use means for genera instead. And there was a discussion which, as long as you thought in terms of classification, which is not the same thing as phylogeny, you couldn't really achieve clarity. And so I was stimulated to work on that in the sort of early to mid 1980s. And I found that there is a problem, and the problem is clearly seen when you look at a phylogeny. And what I want to do is show you it to you in a very simple, rather extreme case. I want to imagine two characters that are, in fact, they don't have any covariation, either selective or genetic. They're independent. They're going to change independently. And here are 22 species. And here's a simple, rather extreme phylogeny that simply has a clade of 11 species here and a clade of 11 species there tied together. And let's imagine evolving two characters that are completely independent of each other in all respects up this phylogeny. And so really all we need to do is take the values for the first character and change them by Brownian motion on this tree, and then do the same for the second character totally independently. And when we do that, we get 22 species out, and if we plot the two characters against each other, the scale here doesn't matter, it's arbitrary. Here's what you get. Here are the 22 tip species. And what biologists who were debating these issues were reacting to was that if you just take those points and consider them as points in a regression, you get, in this case, a nice positive covariation, a positive slope between those two. But if you start taking those points and coloring them in according to where they were on the phylogeny, you get this. You remember that the clades had different colors. One was black and one was gray. Here are all the gray points. That's the gray clade. Here are the black points. And now you look at them and you have to worry, because within the gray clade, there's actually no particular sign of covariation. And within the black clade, there's no particular sign of covariation. But the differences between the clades are resulting in this net positive covariation, the net positive slope. So maybe the slope is due just to the evolution that separated those two clades. And how do we think clearly about this, and how do we make inferences about the covariation of the change of the characters in cases like this? Well, here we have a tree. Now, remember, we were talking about Brownian motion, so we can imagine this is a tree on which things like Brownian motion are happening. although in multiple characters, and those characters show genetic covariations and selective covariation that will cause them to change in correlated ways with a covariance matrix of the changes. Let's think for the moment in terms of Brownian motion, leave Hornstein Uhlenbeck aside. And we might imagine the characters, multiple characters changing on this phylogeny. Now we've seen that just using the tip values is not really a good thing to do. And it's not a good thing to do because the regression machinery that we were using in the previous slide assumes, if you look at the statistics, it assumes that each point is drawn independent and identically distributed from some distribution. That's the common assumption of all kinds of regression and lots of other multivariate methods. Well, if you think about it, the species on this tree are not independent and identically distributed. And in fact, that's the whole point of a phylogeny. A phylogeny is a diagram of non-independence of the resulting species. So when you have a phylogeny, you immediately are not in the independent, identically distributed case. Well, if that's the case, if we're trying to make an inference from non-independent data, how do we do it? What I'm going to be doing is talking here about what's called the contrast method, and it relates to other methods, and Marguerite is going to be talking in the next lecture about that. But let's just think in terms of contrast. You'll remember that I had this rather arbitrary process in which I could take differences among adjacent tips and replace the tips, remove the tips from the tree, and replace them by a weighted average properly computed. Well, if I start to do that on this tree, I'll take a difference between A and B, I'll remove it, replace it by a weighted average. Now we have a tree of 4 species, D and E are over here, and a weighted average of A, B, and C. And the result will be, if I keep doing this, if I keep removing those differences, I'll find that one of them is the difference between A and B, and it has a variance. I didn't say this, but I should have said, when there's the difference between A and B, the variance of that difference is the sum of the variances. Here they're 0.3 and 0.1. To be more correct, in any particular character you look at, it's a constant times 0.3 plus 0.1. So in this case, you have 0.4 as the variance of this difference is proportional to 0.4. The variance of the difference between D and E is proportional to 0.2. If you look on the tree, you'll see they each have a branch length of 0.1. The variance of the weighted average of A and B with C, it will have a variance proportional to 0.975. And finally, when you removed all 3 different contrasts and you're down to a 2-species tree, you have a weighted average of A, B, and C minus a weighted average of D and E. And you can calculate what the variance of that is. So it's possible, when there's a tree, it's possible to get these contrasts by this process of differences and then replacing those 2 nodes, those 2 tips, by a weighted average. Now you have a smaller tree, and you just repeat the process. And what you're doing is coming up with, in this character, values for what are called the contrasts that are independent of each other. They will show no covariation. They are independent, and if you scale each of these differences by the square root of this variance, if you divide the difference by the square root of the variance, you get values that have a common variance. So they're independent. By the way, they have expectation 0, and they have a common variance. So now you have data that are independent, identically distributed. And the interesting thing is, and this I don't have a good slide on, you can do this in any character. So I might be doing it in brain weight. I might also be doing it in body weight. And the covariation between those two traits is still there. After I collect a bunch of contrasts in brain weight and a bunch of contrasts on the same tree in body weight, they covary with each other in the same way they did before. So we could still study the covariation of traits, but now we have independent, identically distributed quantities. We'd sort of factored the tree down into a series of 2 species trees that are independent of each other. And by the way, the number of them is 1 less than the number of tips. So if we have 100 tips, we'll get 99 of these contrasts out. That's assuming, of course, that someone has given you a phylogeny and it has branch lengths, and you're willing to assume that the variance of change along the tree, excuse me, the variance of change along the tree is proportional to the estimated branch lengths from the molecular phylogeny. So now we have a process that addresses the issue of the non-independence of points and replaces the original points by their contrasts. If I do that to the 2-clade example, that one that had 2 11 species clades, the one where some were colored gray and some were colored black, here's what I get for the simulation whose results I showed you. The gray points here are not grade species, they're contrasts. You can get 10 contrasts within the 11 species gray clade, and there they are. And you can get 10 standardized contrasts within the black clade, and there they are. And then there is 1 contrast between the weighted average of the gray and the weighted average of the black, and there it is. And looking at all of those, you can see that it's giving you essentially the right answer. That whereas if you use the TIPS regression, you were completely misled. The non-independence of the points resulted in a spurious positive correlation. Here, there's no sign of the positive correlation. And indeed, we know that in the computer simulation, there wasn't any correlation between those 2 characters. Here's an example. Excuse me. Here's an example of, it's a body size, and let me just see what we have here. What are the characters? Body temperature versus body size in a study by Rieke and Geiser in Biological Reviews a few years ago. Here is the TIPS regression. There's a large number of species of mammals here. I don't know how many, but a great deal. If you just simply take the individual species and you plot body size against, this is log body size because it's often very good with size variables to deal with them on a log scale. And here's the body temperature. We find a cloud of points. There is a regression through them. There is signal of a positive regression. If you treat those points as independent, you get a p-value of 0.000375, which is strongly significant. But watch out, those came off a phylogeny. When they took the phylogeny into account and derived contrast from the phylogeny, you get 1 fewer point, but you get, these points are now contrasts. And when you do contrast versus contrast, we get a non-significant correlation, a non-significant slope. So in fact, this apparent positive slope here is entirely an artifact, or seems to be an artifact, of the non-independence of points. Excuse me, I got a cough for a moment. Just a second here. I actually don't have a cold, but it's something else. It's sinus or something. I don't know. Okay, so there's an example of how taking the phylogeny into account using phylogenetic comparative method, you're able to, using the Brownian motion model, make some inferences about the covariation of, this is the covariance, not the genetic covariance, it's the covariance of changes. It's essentially the covariance of selection response and genetic drift in terms of changes along the tree. You might wonder what you can do to take into account the uncertainty of your tree. Because people are going to give you molecular phylogenies. Those molecular phylogenies are noisy. How can we take that into account? Well, one of the major ways of assessing the noise in a phylogeny is bootstrap sampling, where in the molecular data, you do a bootstrap sample of molecular sites in the molecular data, and you get different trees out of each round of bootstrap sampling. So I'm imagining that somebody has given you a cloud of bootstrap estimated trees. They've given you maybe 100 trees that came out of a bootstrap analysis. And here is just a cartoon of what you could do with that. You could think of doing with that. Suppose we take one of those trees and we use something like a contrast method or one of the other methods that Marguerite will talk to you about. And we assess, we take a bootstrap tree and we use that tree to make an estimate with our morphological data of the covariances of change along the tree. Well, that's just an estimate, and we're doing it not for the true tree, but we're doing it for a bootstrap estimate. Well, you can simply think in terms of doing this separately for each bootstrap tree in the sample. That may seem terribly tedious, but we have computers, and the contrast machinery is actually very quick. It's not computationally all that tedious. So it's quite possible to think of getting not one covariance matrix, but a cloud of covariance matrices. So now, in effect, you have bootstrap estimates of the covariance matrix that you're interested in, the covariance matrix of evolutionary changes. And you've done it by simply using every tree in the bootstrap sample. You can also think about Bayesian approaches. If you have a cloud of trees from the posterior of a Markov chain Monte Carlo Bayesian inference of the phylogeny, such as you might get out of programs like Mr. Bayes, you can think about doing the same thing. In a Bayesian model, you could just do estimates separately on each tree in the posterior sample, but you could also even think about putting the whole process together as a Bayesian process. In that case, we would have a full Bayesian analysis of trees and covariances. We would have a prior on the trees. We would have a prior on the covariances, not a single point covariance, but a prior that tells us that specifies the variation, the uncertainty in the covariance matrix. And here's my silly cartoon of what we would do in a case like that. You also have the molecular data, and the kind of analysis you do in a program like Mr. Bayes deals with these three down in the lower right. But if you were to put the whole thing together as a gigantic Bayesian model, which at this point I don't think people do much, maybe Mr. Bayes has some way of doing this, you could think about predicting the morphological data from the tree and from the covariances. And then you could say, well, let's do Markov chain Monte Carlo on this. Let's start wandering in tree space and wandering in covariance matrix space. And then here's my silly thing. They would wander. There they are wandering. This is Markov chain Monte Carlo. And the result would be you would end up with a posterior sample of your covariance matrices. Now, in practice, maybe people won't mostly do that. With Bayesian data, they mostly will get a cloud of trees out of a Bayesian analysis and then do what I showed earlier, get a separate maximum likelihood estimate of covariances from each of those trees. So that becomes a somewhat Bayesian, somewhat likelihood, kind of weird hybrid. But you could imagine even putting the whole process together in a full Bayesian MCMC under this somewhat idealized model of what the characters are doing and the character covariation. Okay, we're getting there. We're not going to take tremendously longer. Let me talk about some of the uncertain,
Joe, I have a
complications.
On
Is there a? Does somebody say
on
something?
the last thing?
Okay, please.
So you can do that in RevBayes. There's tutorials online that show you how you could estimate both the tree and the covariances at the same time. So that is implemented in RevBayes. There's scripts to do that.
I'm not hearing clearly. You say you cannot do it, or you can do it?
You can do it. You can do it. It's implemented in RevBayes, so you can do that right now in RevBayes.
I still cannot hear, the sound is not good coming from the room. I cannot hear whether you're saying can or can't.
You can. RevBayes can do everything, including what you just said, apparently.
And it will do it, but presumably there is an issue of what's the prior on the variation of covariance matrices. They will have made some choices. You have to be, I would be very alert about the implications of the choices they made. So, okay, if that would be great to be able to use Mr. Bayes to do that analysis and then get out a cloud of covariance matrices, and that will give you a very real sense of the statistical noise that results from the uncertainty of the tree and also from the measurements that you made on the quantitative characters. Great. All right, I was going to list some covariances, some complications here. We dealt with the issue of uncertainty about the phylogeny. There's a whole separate issue of small sample size. I have been, in everything I've said so far, I've naively assumed that we have a species at the tip of the tree, well, a bunch of them, and that we have measured the mean phenotype for our characters in those species. Well, of course, that's naive. We have a sample. We might have only 5 museum specimens. So small sample size is an issue. Robert Ricklefs and Stark in 1996 raised this issue and said, hey, folks, this is a problem and it's biasing our results. I won't go into details, except to say that Ives, Midford, and Garland in 2007 came up with a way of dealing with that. If you knew the phenotypic covariances of the individuals, and I came up the next year, published a way of dealing with it where we estimated the covariance of change of the characters and the phenotypic covariances for the sampling within species. So in my case, you estimate them both off in the analysis. Ives, Midford, and Garland assumed that you knew one of them. So there are these ways of dealing with small sample size, and you have to be, I would urge people to be sensitive to that issue. I have been acting as if the branch links on the molecular tree tell you about the scale relevant to the change of morphological characters. in effect, I'm not assuming a morphological clock, but I'm assuming that both morphology and molecules are changing in a non-clock-like fashion proportional to each other. That's a rather arbitrary assumption, and it needs to be worried about. Nobody's doing much about it, but the one way would be to say maybe there's a molecular clock, we'll estimate a clock-like tree, and then we'll assume also a morphological clock. But again, it's not obvious that that things like correlated morphological characters are going to change in a clock-like fashion. And I should just add that when you do regressions and correlations and other multivariate statistical analyses using contrasts, the nice thing about contrast is under the model, their expectation is zero because you have two tips. You don't know which one's going to have a higher value. A priori, they have the same expectation. So the difference between them has a expectation of zero. So you can do statistical methods under the assumption that you know the mean, you know the expectation, and that's zero. Let's see, what else? Oh yeah. Here now we come up with a big hairy issue. And it involves also the point that was raised earlier about regressing phenotypic values on environmental variables. And I have a major worry on this. And it's not only me. Hansen and Bartoszek in a paper in 2012 raised the same issue. And I think it's one everyone, when you have a research program of relating phenotypes to environmental variables, you have to worry about this one. The phenotypes you see today, let's imagine our environmental variable is temperature. And we have a series of species, and we measure phenotypes, and we measure the temperatures in the present day in those populations. Well, the phenotypes evolved, perhaps they evolved in response to temperatures, but the temperatures they evolved in response to are the ones of the past. And since the tips have different temperatures in those different species, different environmental temperatures, it follows that the temperatures have changed along the tree as well. And we don't have those data. We just have the data about the tips. Present day methods for regressing tip phenotype data on environmental variables such as temperature involve regressing on temperature in a tips fashion, in a non-phylogenetically aware fashion, and regressing on the present day temperatures, and then taking the residuals and working with those. Now, perhaps some of the people who raised this a moment ago could comment on whether I've got that correct, whether that is in fact the general approach. But we don't know what the temperatures were in the past. The only way of doing a fully time-aware and phylogeny-aware analysis is to have a model for the change of the environmental variable as well. So then we might say, temperature is changing by Brownian motion on this tree, the temperature of the environment, and the phenotypes are changing by Brownian motion. At that point, some meteorologist might jump up and say, why do you say Brownian motion? What is it that, why do you assume that your environmental variables are doing the same kind of a process as your biological, as your phenotypes of your species? And the embarrassing answer is, well, for convenience, it's tractable, so we'll assume that. So I think there's a literature coming in which people will not just try to measure present-day environmental variables on individual species and correct for that, but we'll be using a model of change of those environmental variables. And that's going to lead to a lot of agonizing about whether they've got the right model for the environmental variables. So I just wanted to point out this problem and point out that many of the phylogenetic regression methods where you're regressing on environmental variables, I think are questionable, and I think they have to be rethought. And I think Hansen and Bartoszek agree on that. Yeah, and that's the point I'm making in the bottom here of can you assume that the environmental variables are doing the same kind of thing as your phenotypes? Okay. One thing one should say as a result of all this is you end up with something that looks very satisfying. It's an assessment of the covariances of the change of all your characters. So now you know for, brain weight, body weight, lengths of parts of the body, you know how they're covariating. You have an estimate of the covariation. It's worth noting, and your statistical methods will tell you this, that these are have very noisy covariances. So often people will come and say, well, I've got 5 species and I want to do a comparative methods analysis, a phylogenetic comparative methods. And I'll say at that point, well, you're going to get estimates of the covariance, but they're going to be horribly rough. They're going to be horribly variable. And people may say, so if you have, well, here, say 50 species sampled, you'll be estimating covariances based on 49 points, the contrasts. And other comparative methods that you do will have a comparable error. Now, we've corrected for the non-independence on the tree. If you are working not with continuous measurements, but with thresholded 0, 1 traits, life gets even harder. And we're facing the problem. The problem we will face is that we can make rough estimates of the covariation across in species evolving on a tree. And we're doing so for an audience, particularly the popular science press, that wants perfection. It wants a fly-on-the-wall certainty. It wants to know exactly what happened in evolution. And I think looking at data on present-day species, even when you know the phylogenies, it's not so easy to get that. So you might say, well, the way out of that is not to take 50 species. Let's take 200 species. The problem is we've been making a very naive, simplified Brownian motion model of our character. As you expand to more species, you're now expanding to species, you're bringing in other groups nearby. They have somewhat different biology. The biology of those same characters in those other species starts to differ. And so the model starts to be at risk. You can expand to more species to get a bigger sample size, but only at the cost that your model starts to creak and groan and fractures appear in your model, and you start to doubt whether, in fact, that's a good model. A simple, naive Brownian motion, correlated Brownian motion model is really good over that broad phylogenetic scale. So you might say, well, measure more characters. That doesn't actually help because it just brings in a lot more parameters, more covariances that you have to estimate. So yeah, you bring in more data, but you're bringing in even more new parameters to estimate, and I think the result is not going to be helpful. Okay, so that's a quick tour of some of the issues in phylogenetic comparative biology. And in using things like Brownian motion models, I've mentioned the whole selective covariance issue. I mentioned a whole bunch of other problems. Let me just show you the references that I have on this, and then we can have some more discussion. Here is papers on on genetic drift, on the population genetics of genetic drift. I have some on the papers that I've mentioned on the use of the Brownian motion approximation for gene frequencies. There's a literature on how you get likelihoods for the trees. We didn't talk about that because we're assuming the tree is given to you by molecular data. There's some brilliant work by my colleague Elizabeth Thompson and some work by me on that. And I have references on multivariate Brownian motion. You'll see some important papers by Ross Landy there. And I even have Olaf Tedin's paper on selective covariance. So, and then the phylogenetic comparative method literature and some of the papers that I've mentioned here.